Okay, good afternoon, everyone. I hope that you and your families are doing well at this time. If you would please, if you can hear me okay, please type yes into the chat window. Okay, great, great. Thank you so much. All right, gang. Well, as I said, I hope you're doing well. I know everybody's getting a little stir crazy right now, but hopefully we'll be better off for it. So, Um, let me see a couple business items. I wanted to mention to each of you the homework assignment that you submitted recently with the programming challenge. I realize that many of you did struggle with the programming challenge, and it turns out that there was some sort of bug in the Wolfram systems at that time that it was not capable of downloading the amino acid data that was needed for many of you. So, please be advised that that was not your fault. You didn't do anything wrong. And I have submitted a bug report to Wolfram so that they know to look into that and correct that problem. So no problems there. Again, that was not your error. It was the company. So, okay, this Thursday, you have your second lecture exam. You know about that. The lecture exam is of course, going to be online using the Classmarker website. And you will have from 7 a.m. till midnight to take the exam. So you can take the exam anytime on Thursday, okay? So there will be no lecture on Thursday, okay? You don't have to Zoom in at 12:30 like you normally would, okay? Because Thursday is just about you taking your exam. So Thursday, take your exam between 7 a.m. and midnight, but there is no lecture this coming Thursday. As you prepare to take your exam, you want to definitely be sure that you're ready to take the exam, that you can put yourself in a room free of distractions. You're not going to be distracted by anybody else you're living with, because once you start that exam, you cannot pause it. You cannot stop it. You cannot come back to it. You have to finish it in one sitting. So when you start it, you must finish it. So please be sure that you are ready to devote an hour and a half to the exam and that your family knows not to interrupt you during that time. Okay, do you have any questions right now about what's going to happen on Thursday in terms of the process of taking the exam? Or is there anything you'd like to talk about in terms of the exam? Okay, Sean's question, once we start, how long do we have to finish it? I think in the study guide, Sean, I mentioned an hour and 25 minutes, but I'm pretty confident at this point that I'm going to extend that to an hour and 30 minutes. So expect that you will have 1 hour and 30 minutes to take the exam. John asks, do we get sent a link or just log into the website and it will appear? Just log into the website, John, and it will appear on Thursday to you. If you folks have any difficulty with the website, God forbid you're taking the exam for a half an hour and your computer crashes or something like that, don't freak out. I mean, it will be upsetting, but reboot your computer, send me an email, tell me what's going on, and I will reset the exam for you. And you can continue from that point. Okay, so any other questions? Okay, so if they come up before Thursday, by all means, shoot me an email. Again, make sure that you have an hour and 30 minutes of undistracted time set aside. Make sure you have a calculator sitting next to you. Make sure you have some scrap paper that you can jot down some notes and work out the logic of problems, particularly the essay problems. And make sure that you've had something to eat so that you're not hungry when you take the exam, because you're going to need some brain glucose to do some thinking. Okay, so let me know if something comes up in the next 2 days and we'll work through it together. Okay, so we're going to start chapter 11 today, folks, okay? Now remember, Chapter 11 is not on your exam Thursday. Chapter 11 will be the first chapter that will be on your final exam at the end of the semester. So Chapter 11 is not on your test on Thursday. So certainly you want to take notes and try to pay attention, but you don't have to study that material for Thursday's exam. Okay. Bear with me a moment, folks, while I go to the Word document and get set up here for you. Okay. So you should see a Microsoft Word file at this point on my screen. And I'm going to bring the chat window over here so that you can communicate with me while we're talking about these topics. All right. Okay, gang. So the topic of Chapter 11 is gene mutation, DNA repair, and transposition. Okay. Okay. So we're going to start talking about mutation a lot today. Now, mutation, okay, so, okay, mutation simply refers to a change in our DNA. Okay, this refers to a change in the DNA of a cell. Okay, many of you I know have not had microbiology at this point, but you should appreciate, and I think you generally do, that mutation really is the driving force for evolution, right? All the different varieties of life on our planet, all the species that are on our planet, well, presumably at some point in our geologic history, potentially billions of years ago, we all evolved from a common ancestor cell. So over the billions of years, mutations have developed and we've turned into different kinds of cells from fish cells to dog cells to elephant cells to bacteria to human cells, right? All life on the planet, as far as we can tell, is related in some way to a common ancestor a long, long, long time ago. Okay. Mutation is the driving force of evolution on our planet. Okay. Certainly, our species, our collective species, Homo sapiens, we are not going to be the same as a species after we come through the other end of this coronavirus pandemic. Okay, our species is going to be forever changed because of how this virus is invading our cells. Now, I don't have time to give you a lecture on exactly what viruses can do to us, but you can appreciate certainly that viruses, some viruses very much have the ability to change our DNA. So let's think about what's happening on the planet right now. We have humanity being infected with this thing called a coronavirus. The recent research that's been published out there suggests that the coronavirus that's currently making people sick is capable of making people sick because the virus itself experienced a recent mutation, which allows it to bind to a receptor on the surface of human cells stronger than it had previously done. In other words, in the past 10 years, coronavirus was not all that dangerous to humanity. Sure, it was circulating around the globe, but it didn't cause a great deal of heartache like it's causing right now. But something in the past 8 to 10 months has changed the virus. It has experienced a mutation so that it is capable of binding what's called an ACE receptor, ACE, an ACE receptor on the surface of human cells. ACE is short for angiotensin converting enzyme receptor, and you may know if you've taken anatomy and physiology that angiotensin is a hormone. So the virus has experienced a mutation that allows it to bind to a hormone receptor on human cells much more tightly than it was able to do before. And that allows the virus to infect our cells more efficiently than before, and is in fact causing a great deal of problems for us. Okay, so recent example, today's coronavirus pandemic is possible because the virus recently, let's say in the past 10 months, has mutated in a way that allows it to bind to receptors on the surface of human cells more tightly than it previously could. All right. So mutation is definitely the driving force of evolution on our planet. And at least in this case, which is very real to all of us at this time, this mutation is resulting in a great deal of suffering. Thoughts or questions, gang? Okay. Well, as you know, we're in class. This is school. This is academic. So we're going to talk about a lot of different things from chapter 11. If you look on page 304 of your textbook, the conversation begins. Okay. Column 1 there says gene mutations are classified in various ways. Then if you look at the right column of page 304, you see the classification based on a type of molecular change. Page 305, you see classification based on phenotypic effects. Page 306, you see classification based on location of the mutation itself. I'm not going to be quite that academic in my coverage of these things, and I'm not going to list every variety of mutation your textbook gives you, but there are certain basic forms of mutation I definitely want to give you notes on and put into your notebook. So let's start with that. Types of mutations. All right, and many of these things you probably are familiar with. I mean, certainly a good Biol 101 and 102 course should have introduced you to these ideas. So let me mention a point mutation. A point mutation refers to a single nucleotide change in the DNA. This refers to a single nucleotide change in the DNA. Now, we might think that that is often innocuous, that a single nucleotide out of billions and billions of nucleotides, you know, for the most part, is going to be harmless. And that is often quite true. But an exception to that, a scenario where a single nucleotide mutation is not harmless, is in sickle cell anemia. Okay. Sickle cell anemia, as you may recall, is a disease of the blood system where the red blood cells take on an abnormal shape. They take on a crescent moon or sickle shape. And that's because the hemoglobin molecule inside the cells is mutated. And the reason that's mutated is because there is an individual single nucleotide point mutation in the gene that codes for the globin portion of hemoglobin. Results in a abnormally shaped cell. It looks like a crescent moon. This results from a single nucleotide change in the DNA of the gene that codes for the globin protein that is part of hemoglobin. Okay, I will slow down so that you can finish your copy. Sickle cell anemia results in an abnormally shaped cell. It looks like a crescent moon. This results from a single nucleotide change in the DNA of the gene that codes for the globin protein that is part of hemoglobin. Okay, question coming through the chat window. Why is sickle cell anemia painful? Do you have any thoughts on that, gang? Do you know why it's painful to be a sickle cell patient? Please type in the answer to the chat window if you know, or if you want to speak up, go ahead and unmute your microphone. Not enough blood flow to the body parts. Well, Hayden, that is a true thing. In other words, there is something going on with blood flow. Can you be any more precise about that? Okay, blood clots. Okay, that's not bad. Ian, the heart has to overcompensate. It does put stress on the heart, but that's not what I'm referring to. Okay, gang, let me type this question into your Word document. Okay, Sean, maybe something with lactic acid and anaerobic respiration. Those processes are definitely impacted. I can't say no to that. But here, question. Why is sickle cell anemia painful? It comes down to this, gang. Because the red blood cells have the wrong shape, and you may recall from your knowledge of human biology that red blood cells pass through capillaries, single file, one after the other. And when the cells have an abnormal shape, they actually catch on each other, kind of like molecular Velcro, so that you do start to form tiny little blood clots in the capillary beds, Hayden. And those tiny little blood clots do block blood flow. But what happens is that the blood cells start to back up in the capillary vessel, which causes the structure of the capillary wall to stretch. And as it stretches, as it gets too stretched, that causes nerve pulses to the nervous system, which are interpreted as pain. Okay, so red blood cells get, think of a famous football play where you have 10 football players laying on top of each other. You have a pile of red blood cells that literally get stuck in the capillary wall. Red blood cells get stuck as they try to move through the capillaries. This causes the capillary to swell, which sends electrical signals to the nervous system that are interpreted as pain. Okay, and of course, if this goes on for long enough, it is possible that the capillary wall can rupture, and all of a sudden we have internal bleeding. Not a good thing. Okay, so, maybe another day, another time, we could talk more about sickle cell anemia, but all of that harm, all of that pain, all of that suffering is caused by one individual nucleotide mutation in the globin gene. Okay, so let me give you an example here of what a point mutation could kind of look like. So let me see here. And this would be better on a whiteboard, but let me do it this way. AATCGG. And then what I want to do is put below it a comparable sequence where you can hopefully, because we're comparing them top to bottom, you can in fact see the mutation. AA, GC, GG. If you compare the top sequence to the bottom sequence, you can see that they are in fact identical, except for one nucleotide. If we start counting nucleotides from the left and go to the right, please type into the chat window, which nucleotide position has the mutation? Okay, thank you, John. I just want to be sure that you're with me, folks. Exactly, Caitlin, you got it. The third nucleotide position is the change. That would be an example of a point mutation. Later on in our lecture here, we will talk about, what causes the mutations to actually form. We are going to talk about that, but right now I'm just going to list out some common mutations that cells can experience. Okay, please feel free to type into the chat window any questions you might have. A missense mutation is the second kind of mutation that I want to be sure that you're okay with. A missense mutation refers to a mutation, a change in the DNA, that changes the codon of a polypeptide. Okay? This refers to a mutation that changes the sequence of a codon, which impacts the translation of a polypeptide. Okay, so an example here would be something like this, AUGCAC. Okay, now this codes, this top sequence, as we interpret each sequence of 3 nucleotides as a codon, this would be interpreted by the ribosome as methionine, followed by histidine. Okay, those are the 2 amino acids that would be put together here. An example of a missense mutation would be this, AUGGAC. Okay, so you can see here that there is a single nucleotide change at the 4th nucleotide position here, and this would lead to the following amino acids: methionine and aspartic acid. Okay? So it's important that you notice how this missense mutation is also a point mutation, right? Here we have a single individual nucleotide change that in this sequence of nucleic acid, which is RNA because I'm using U's, right? This missense mutation is a missense mutation, but it's also a point mutation. Okay, a point mutation is leading to a missense. It's leading to an improper interpretation of a codon and a piece of messenger RNA by the ribosome. Okay. All right. Any questions, gang? Okay. I will continue to try to be relatively slow in my notes here so that you can keep up. Go ahead, Hayden.
You said you changed the sequence of the codon, but you just changed one letter.
Correct. So let me see here. Right here is the G that's been changed. That one letter on the top sequence is a C, and on the bottom sequence is a G. That single letter change is a point mutation. And so the codon is now going to be GAC instead of CAC.
Okay. But you didn't change the sequence. I thought that meant like reversing it. You know what I mean?
I don't know what you mean.
Okay, so what's the difference between the missense and the point then? Mutation.
Okay, thank you for asking that. Let's put that down here. Okay. So question, is there a difference between a point mutation and a missense mutation? Yes. Okay, remember from our conversation over the past couple lectures, Hayden, that not all of your DNA is translated ultimately into a protein. In fact, very little of it is. Okay, so that means that there are plenty regions of your DNA that don't code for a protein, but still could experience a single nucleotide change. Those would be point mutations. For a point mutation to also be categorized as a missense mutation, the point mutation would have to specifically be in a gene that ultimately is translated into a protein. Does that make sense?
Yes.
Yes, many regions of DNA do not code for protein, but can still experience point mutations. And now remember from previous lectures, we mentioned that a lot of DNA is not translated into protein, but a lot of the DNA that's transcribed into RNA, which then is not translated into protein, that RNA typically has regulatory capacity in the cell. So a point mutation that is not a missense mutation can still screw a cell up because it affects some kind of regulatory information inside the cell. Okay, if a point mutation is inside a codon that is normally translated into protein, then the point mutation is also a missense mutation. Okay. All right. Hayden, are you okay?
Yes.
Okay. All right, gang. Keep firing away. That's why we're here. Let's take a look at a nonsense mutation. Okay. And well, let me back up and say one more thing here under the missense mutation. Okay. Letter 2, letter A. I said, notice how this missense mutation is a point mutation. Not all missense mutations have to be point mutations, right? Could not it have been that I changed both the C and the A to something else? Maybe I could have made this GCC, right? So in that case, then that missense mutation was not due to a single point mutation, but by the 2 point mutations that happen to be next to each other. Okay, so let's put that up here. Notice how this missense mutation is also a point mutation, but not all missense mutations are due to individual point mutations. Okay. All right. Continuing, nonsense mutation. A nonsense mutation is a mutation that changes the codon to a stop codon. This is a mutation that changes a codon to a stop codon. All right. So as an example here, we could have GGACAC, and that would code for glycine and histidine. But if I have a nonsense mutation, I could end up with UGA, CAC. And in that case, the first codon here, the UGA, is actually a stop codon. Okay, so that's stop. And since CAC is downstream of the stop codon, CAC would never be translated into an amino acid. CAC is not translated into an amino acid. Okay, so you can see how that could severely disrupt the size of a polypeptide, the size of a protein that might be made. Number 4, a silent mutation. Okay, a silent mutation. Okay, a silent mutation refers to a change in the nucleotide sequence that does not lead to a change in the amino acid. Okay, there's a change in the DNA or RNA. Well, obviously, you're going to have a change in the DNA, which leads to a change in the RNA. But then when that RNA is translated, it does not lead to a change in the polypeptide. This refers to a change in the DNA, which then changes the RNA, but the polypeptide remains unchanged. Okay. All right. So as an example, well, before I give you an example, I want to see if you understand what's happening here. Okay. So I have a question. I'll put the question into the chat window first. What characteristic of the genetic code allows for silent mutations to exist? What characteristic of the genetic code allows for silent mutations to exist? Please type in the answer if you know it. Okay, John wins the Cupid doll. Very good. Thank you, John. Exactly. Different codons can equal the same amino acid. Different codons do equal the same amino acid. Let's see here, one moment. Let's see here. If you take a look, okay, so let's write down the answer to that, and then I'll direct you someplace else. What's characteristic of the genetic code allows for silent mutations to exist? And John said it, multiple codons can specify the same amino acids. Okay, so John had the right answer. Now my next question coming through the chat window, the fact that there are multiple codons that equal the same amino acid, there's a special vocab word that describes that. What is the vocab word that describes the fact that multiple codons specify the same common amino acid? What's the vocab word that represents the answer that John put down in the chat window? Okay, degenerate, fantastic. Okay, good. That's what I'm fishing for. Very good, gang. Okay. So, multiple codons can specify the same amino acid. This is known as degeneracy. Okay, very, very nice. Okay, so let me give an example of that. An example of that would be GGACAC. That codes for glycine and histidine. And the following nucleotides code for the exact same thing, but they are GGC, CAC. Okay, so that's also glycine and histidine. But you can see, I know that the fonts aren't an exact match, unfortunately. You can see that the first codon on top is GGA, the first codon on the bottom is GGC. Well, both of those codons refer to the same glycine amino acid because the genetic code is degenerate. Okay. And so hopefully that's something you picked up on when you were reading from chapter 9, page 244. Figure 7 has an example of the genetic code. So just to be sure, if you have your textbook easily accessible, I hope it is in front of you, please turn to page 244. And my next question is coming through the chat window. How many, hold on one second here. This genetic code table made it too easy. How many codons, how many different codons specify leucine? How many different codons specify leucine? Okay, thank you, Ian. Ian's got it. It's 6, right? There are 6 different ways. There are 6 different ways. 6 different ways that a messenger RNA can specify glycine. Okay. Now there is one small talking point I have to give you right here, and that is this. Silent mutation means, from a genetic perspective and what it has come to mean over the many, many years of genetic research, silent mutation has come to mean that there is a nucleotide change that does not get reflected into the protein that's made. So therefore it seems silent. But what we are beginning to understand is that even silent mutations, while they may in fact not change the protein that's made, the silent mutations can change other subtle aspects of how genes are expressed. And it's something we're just beginning to appreciate. Okay? Even though silent mutations do not alter the protein, science is beginning to appreciate that there can be other ramifications to gene regulation and gene coordination, gene expression coordination from silent mutations. Okay. So there can be some things happening under the covers, behind the canvas, in ways that we are not yet completely understanding with the silent mutations. Okay, questions at this time?
So tell me if you can't hear me, but if that means that there could be changes that we don't know of, does that mean that, like, in regular DNA that isn't mutated, that's correct, like if there's 6 different codons, do each of those 6 different codons have a different regulatory function for lysine?
John, the quick answer to your question is that they probably do. Okay? While the genetic code table you see in your textbook is pretty universal for all life on the planet, it is certainly true that different species of cells prefer to use specific codons. Just as a random example, E. coli bacteria may prefer to use UUA as its codon for leucine, while as human cells might prefer to use CUG. I'm not saying it's that way literally because I don't have that memorized. But what I'm saying is that different kinds of cells on the planet do have specific codons that they prefer to use for specific amino acids, even though all life on the planet can collectively use multiple codons to specify a single amino acid. Does that make sense? Okay. All right, John. Okay, let's keep going, gang. Number 5, frame shift mutation. Not frame shift, frame shift. Okay, frame shift mutation. Okay, this is a kind of mutation which results from the insertion or deletion of nucleotides which alters the reading frame of the polypeptide that could be made. This mutation results from an insertion or deletion of nucleotides which alters the reading frame of the polypeptide that is made. Okay, let me give you an example here. Well, we could have GGA-CAC. This typically codes for glycine and histidine. And then what we could do is have the following. We could have GCC, GACAC, okay? So we've taken the top sequence of 6 nucleotides and turned it into a sequence of 8 nucleotides because of an insertion, right? Because of an insertion. Now this, okay, this would code for okay, so the first codon up top was GGA that coded for glycine, but now because of the insertion, right? Notice that these 2 C's represent an insertion because the next nucleotides are identical to what's found in top. So these 2 grayed out nucleotides right now are the actual insertion. And because of that, we would end up having the following amino acids in our protein. We would have alanine, followed by aspartic acid. And then there would be 2 nucleotides left over that presumably nothing happens with it. Okay. All right. Now, in this case, you know, when I'm able to give you some real world examples of things, I like to do so. In this case, I'm trying to think back to my microbiology days. There is at least one commonly used antibiotic. I think it's either tetracycline or streptomycin. I'd have to look it up, but it's one of the historically common antibiotics that actually causes a frame shift mutation in the bacteria as they are manufacturing proteins to make their ribosomes. And when that happens, the proteins to make the ribosomes are not made properly, so the bacteria can't make their ribosomes properly, and so the ribosomes don't work properly, so the cells actually die. Okay. And at least one famous antibiotic against bacteria, and it's probably streptomycin or tetracycline. I'd have to look it up. Causes a frame shift. Damn, I want to say shit. Frame shift mutation that prevents bacteria from making competent ribosomes, so the bacteria die. Okay, so this kind of mutation, like all mutations, can have wide consuming consequences. Okay, any questions right now, gang? Okay, so hopefully, hopefully 6 examples of things are enough that you get to see how dramatic the effects of mutations can really be. I'm going to give you a couple other definitions for mutations, but we're not going to belabor the point too much here. Let's talk about 6, a loss of function mutation. Okay, well, this is exactly what the phrase suggests. This is a mutation that reduces or eliminates the function of the gene product. Okay, maybe you have an enzyme that, due to mutation, isn't as efficient at breaking down its substrate anymore. Okay, any mutation that reduces or eliminates the function of the gene product, that's a loss of function mutation. We have recessive mutations. Okay, these are mutations whose phenotypic impact is typically masked or hidden in a diploid organism by the presence of a wild type allele. A mutation whose phenotypic impact is masked in a diploid organism by the presence of a wild type allele. I'm going to give you 2 more here, and then we're going to move on to talk about some other ideas. Okay, number 8. Well, if there's a recessive mutation, you can bet there can be dominant mutations. Okay, this is a mutation whose phenotypic impact is observed in a diploid organism, even in the presence of a wild type allele. Okay, a mutation whose phenotypic impact is observed in a diploid organism, even in the presence of a wild type allele. And then the last example I'm going to put into your notes is a temperature sensitive mutation. Okay, this is a mutation whose impact or consequence is observed at one temperature, but not the other. I'll give you an example of this in a second. Whose impact or consequence is observed at one temperature, but not another. Okay. Perhaps some of you, and now I know everybody's thinking about coronavirus right now. That's on everybody's mind at this time. But before this emergency came up, we would typically battle the seasonal influenza virus that created flu, and flu itself is no joking matter because that kills tens of thousands of people every year around the world. Okay, so you may have gotten a flu vaccine, or you certainly are aware of the existence of flu vaccines. Now, there's different versions of flu vaccines that are out there, and I don't know, I'm ballparking this, but maybe about 10 or 12 years ago, a new version of flu vaccine was initially put on the market called Flu Mist, where rather than getting an injection, you would actually inhale it in the nostril, and then through a consequence of series of events, you would develop immunity from this vaccine. Folks, let me ask you here, have you heard of flu mist? Okay, Ian has. All right. How about the rest of you? Okay. All right. All right. So let's talk a little bit about this. Okay, good. Okay. So some of you haven't heard about this. About 10 years ago, a new version of flu vaccine was developed called flu mist. Okay. This was an inhaled vaccine. It was not an injection. And obviously the benefit of that is for people that are very scared to get injections, you didn't have to get one. And so this was pretty good, particularly for children, because, you know, children don't like needles. Well, long story short, the basic way that this worked is that this flu vaccine contained a genetically modified version of the flu virus that was able to infect and grow and replicate in cells of your sinus cavity. But as the virus began to move deeper inside your chest cavity, the temperature inside your chest cavity is different enough, even by just 1 degree, it's different enough that the protein molecules of the flu virus would change their shape and deactivate. So you would inhale this flu mist vaccine. It would contain a live virus that infected your cells in your nasal cavity. The virus would replicate. That would agitate your immune system, so your immune system would begin to fight it and develop antibodies against it. And then as that flu virus began to move deeper into your body, because of a literal change in temperature between your nasal cavity to the internal thoracic cavity of your body, the flu virus would deactivate and not be a threat to you anymore. But because your immune system got riled up by what was happening in the sinuses, in the nasal cavity, you did develop protective immunity against the virus. Okay, am I making sense? Okay, I'm not getting a response, so I'm hoping I'm making sense. Okay. In this case, let me see if I can put this down in as few words as possible. A genetically modified virus is inhaled, and the immune system is activated as the virus replicates in the nasal passageways of the body. As the virus spreads deeper into your body temperature changes enough that the molecules of the virus change shape and deactivate, meaning the virus cannot replicate anymore. So the virus is not a threat to you, but your immune system was in fact turned on enough to create protective immunity.
So they mutated it to die at a certain temperature.
That is correct. Okay. The virus is not a threat anymore, but your immune system develops protection for you. This is a beautiful example of a temperature-sensitive mutation that led to a real-world product called flu mist. Now, as life has it, you know, working with biological samples is difficult. Okay, Mother Nature doesn't want to play well with you. So what actually ended up happening about, I don't know, 5 or 6 years ago, is that the companies that were making this flu mist realized that the flu mist wasn't protecting people as well as they thought it was going to protect people. So they pulled it off the market for a year or two. The FDA pulled it off the market. And long story short, a subsequent analysis of the flu mist virus showed that the virus had actually mutated, and it had mutated in such a way that your immune system wasn't creating the immune response that we needed to protect against it. So that's the difficulty dealing with sort of living biological products, is that the thing that you're using as a product doesn't care that you're using it as a product. It's simply a living thing. It's going to do what it does. In that case, the virus had mutated and the product wasn't as effective as it had been. Subsequent to that, the companies that make this have altered the virus again, and it's back on the market as far as I know. So hopefully it'll be back on the market for a long time, but it doesn't preclude the possibility of the virus mutating again a second time and screwing things up. And that's just how it is when you're dealing with living medicine. Okay.
I had a
Questions?
question. So do all of the virus cells of that specific virus mutate in the same order?
No. Let me say a few things. Forgive me, I'm a teacher, so I got to pick on you. Okay, viruses are not cells. They are not cells, so please don't call them virus cells. They are not cells. Viruses are simply particles that infect cells. But at any rate, the answer to the question is no, viruses do not mutate in the same way. Let's say you got infected with one virus particle that caused you to have a cold. After 10 days of having the cold, if you were to harvest all the cold virus particles in your body, you would find an entire spectrum of different mutations in that original cold virus. Does that make sense?
Yeah. So how do they figure out what mutation to change? Because if somebody else had the same virus, they would have a whole set of different mutations.
Correct. So typically what a company will do is look at statistically what the most common changes were and make adaptations for those most common changes.
Makes sense.
Anything else, gang? If you realize that when you get infected with a virus over 2 weeks, you literally have a civilization of slightly different viral variants in your body, it's even more impressive when your immune system is able to tackle them enough so that you don't die from it. I mean, it is amazing what your immune system is able to do. And it's also not too surprising when our bodies become overwhelmed by these pathogens and we succumb to them. Okay, let's keep going, gang. Next, I want to shift our conversation in chapter 11 to the nature of mutation itself. Kind of have a little bit of a philosophical talk here. Okay. So I'm looking at information on page 306 and 307 and 308 of your textbook, 306, 307, 308. Okay. First off, the experiments that scientists have performed on mutations have suggested that mutations are often random in nature. Okay. In fact, I think your book hits that a little bit too hard because it tries to emphasize that mutations are random, or in the language of your book, spontaneous. They just pop out of thin air. Well, that is possible. Keep in mind that just because something's random does not mean, if something is random, it's not predictable by us. We can't predict it. But that doesn't mean there isn't an explanation for it, right? Having something be random doesn't mean there is not an explanation. It just means we can't predict what happened. So what I'm trying to suggest to you is that, there are many scientists that accept the possibility that mutation is often intentional or induced. They are adaptive in nature, okay? Okay, many scientists believe that at least some mutations are intentional, which is to say that mutations can occur intentionally to allow for some type of adaptive response to an environmental pressure or trigger. Okay. So keep in mind that while many mutations can in fact be unpredictable, they can be random. It doesn't mean they aren't intentional. They may have been done intentionally to do something that we don't understand. Okay. All right. Number 3. On page 307, you have table 1. Okay. I'm just pointing out because I want you to notice the mutation rates that are listed in that table. Okay, so let's define what a mutation rate is. Okay. Mutation rate is the likelihood, and we could replace the word likelihood with fraction. We could replace the word fraction with probability, okay? Mutation rate is the likelihood or fraction or probability that a gene will undergo a change, meaning a mutation, in a single generation of cell division. Okay, so right, if we're talking about bacteria, that cell division is going to be binary fission. If we're talking about human cells, it's going to be mitosis or meiosis. And we've covered that fairly extensively. Okay, so mutation rate is the likelihood that a gene will undergo a change in a single generation of cell division or in the formation of a single gamete. Okay. All right. So this opens the possibility of doing some actual basic reasoning and computations. I want to challenge you next with a question. Okay. All right. That question is this. All right. I'm going to give you a question in relation to the E. coli data on table 1 in relationship to the lactose fermentation character, where you have lac minus going to lac plus at a rate of 2 × 10⁻⁷ per cell division. So here's my question to you. If we start with one bacterial cell and allow it to reproduce into a population of 1 billion cells, how many mutations do we expect to find at the lac locus in bacteria if its known mutation rate is 2 times 10 to the -7th power mutations per cell division. Okay? If we start with one bacterial cell and allow it to reproduce into a population of 1 billion cells, how many mutations do we expect to find at the lac locus? In other words, how many bacteria in that population of 1 billion are going to have a mutation at the lac locus if we know that the mutation rate of that gene locus is 2 times 10 to the -7 mutations per cell division? So first off, is the question making sense? Read it a couple times and see if it makes sense. I'm not getting a response. So do I need to restate this a different way for you? All right, let me see if I can clean this up a little bit. Thank you, Ian. If we start with one bacterial cell and allow it to reproduce into a population of 1 million cells, how many cells do we expect to find a mutation at the lac locus in bacteria if its known mutation rate, if the mutation rate of it being the lac locus, if its lac locus known mutation rate is 2 times 10 to the -7 mutations per cell division. So John asked, so 2 times 10 to the 7th mutations happens 1 billion times? No, so 2 times 10 to the -7th mutation per cell division. Per cell division, John. So I'd like to give you a couple minutes to try to puzzle away at home. See if you can answer this. Okay? See if you can answer this question. I'm going to give you, how about this? I'll give you 3 or 4 minutes. If you think you have an answer, then type in "my answer is" into the chat window and we'll see how you folks do. Go ahead, see if you can figure this out. Sean, thank you for your answer. It is incorrect. Ian, that's incorrect. Okay. Okay, so in about a minute, I'll give you folks a hint, but I want to see what you can do here. Malachi, all right, that's not right, but thank you for trying. Okay, so let me type a hint into your Word document here. To answer this question successfully, you have to be able to figure out how many cell divisions does it take to create a billion cells. Right? We're starting with one cell and we're finishing with a billion cells. Well, how many cell divisions does it take to create that billion cells? Oh, my bad on that. Malachi, I was too quick on that. Malachi, you are right. Malachi, you are right. Okay. So Malachi says 200. How about the rest of you? Are you able to reproduce that answer? Do you know how Malachi got 200? John says because we take the mutation rate times the number of cells. No, you're not taking times the number of cells. You're taking times the number of cell divisions. There's a difference. Caitlin got it? Fantastic. All right. All right, gang. We are getting a little long in the tooth here. So let's be sure we're all okay here. So can anybody tell me, go ahead and type it into the chat window. And Malachi, I apologize. I said you were wrong and then you correct. You are correct. Folks, how many cell divisions does it take to create a billion cells? I'm going to type that into the chat window. How many cell divisions does it take to create 1 billion cells? Okay. Looks like, okay, thank you, Hayden. That is not correct, but I appreciate your trying. Okay. Folks, we're getting a little long in the tooth here. Let me, anybody else have a guess on the number of cell divisions to create a billion cells? Malachi, you must have had it right if you did this. Okay. Folks, at this time, what I'll do is go to a PowerPoint drawing just so I can draw a little bit for you to help you answer this question, and then we'll call it for today, okay? So bear with me. Okay, so now you see a PowerPoint up on the screen, and the question we're dealing with is how many cell divisions does it take to create 1 billion cells? So if we start doing a little drawing here, we can make it easier on ourselves. Okay, so, oop, and make it easy on myself too if I click the right button. Okay, so if we start with a single cell and it reproduces itself, we end up with 2 cells, right? And then each of those 2 cells reproduces into another 2 cells. And each of those cells divides and becomes 2 cells. You get the idea of how this would continue on infinitum until we get to a billion. Okay. So let's think about it. Right here in this row, I have 1, 2, 3, 4. Excuse me. I have 1, 2, 3, 4, 5, 6, 7, 8. So at this level here, I have 8 cells. And it took, hold on, let me change my highlighter here. It took 1, 2, 3, 4, 5, 6, 7 cell divisions to get those 8 cells. That pattern is going to be true the whole way up to 1 billion. So if we have a billion cells, how many cell divisions did it require? Go ahead and type the answer into the chat window. There you go, Ian. 999,999,999 cell divisions to produce a billion cells. So at that point, then, if you want to answer this question, all you need to do, thank you, Caitlin, all you need to do is take the mutation rate, which is 2 times 10 to the minus 7 mutations per 1 cell division, and you multiply that by the number you just calculated, 999,999,999 cell divisions, right? Our units cancel out beautifully, and we end up with, I think it comes out to be like 199.9 or something like that. So 200 mutations. So what we're saying is that we would expect 200 of the cells to have a mutation at the lactose gene locus. 200 cells have a mutation at the lac locus. Okay. All right, gang. Are you okay right now? Any questions? Do you want me to go back to the Word document or anything? Okay. All right. So, folks, I'm going to stop sharing. All right. Okay. So, Thursday, you have your exam. Please log into the class marker website anytime between 7 a.m. and midnight. If you have any technical problems, send me an email. I'll work to get that fixed for you. Once again, there is no lecture on Thursday. Please don't try to log in at 12:30 on Zoom because I won't be online. There is no lecture Thursday. All you have to do on Thursday is do a real good job on your genetics exam. Gang, do you have any questions?
So I missed the first part of when you were talking about the exam. Could you go over the other details you were talking about other than that it's from 7 a.m. to midnight on class marker?
Yeah, I don't think you missed too much, Hayden. It's from 7 a.m. to midnight on class marker. Once you start the exam, you cannot stop. You cannot take a pause in it. So you got to be ready to work for an hour and a half to get through the exam. And I recommend that, you tell your family members and so forth, they've got to leave you alone for an hour and a half, because you really need to focus for an hour and a half. Make sure you have your calculator next to you and a bunch of scrap paper that you can write notes on and jot ideas down to help solve problems, logic problems with the essays. But that's about it.
Okay. Is each question timed individually or the whole test is timed
The
together?
whole test is timed. You can choose to spend as much time on any question that you want, but you only have an hour and 30 minutes to do the entire exam.
Are we allowed to go back to questions?
Yes, you may. Yes, you may.
Okay.
There are going to be buttons in the web page that allow you to go back and forth through the questions.
Okay.
Okay. Anything else? Okay, so don't let the website throw you for a loop. It's just technology. And if there's a problem that comes up with the website, just shoot me an email. We'll get you straightened away. Just do your best to do well with the genetics exam. Okay, so folks, I will stay online. About 2:05, I'll be back online here. If any of you need to chat with me, I'll be happy to do that. Otherwise, I won't see you again until next Tuesday. And so please be safe and take care of yourselves and your family. And I will see you later.
Before you leave, I just want to double check. It's chapter 7 and 10, right?
The exam?
Yeah.
Exam 2 is chapters 5, 6, 7, 9, and 10.
Okay.
Okay? All right, gang. Take a break. If you need me, I'll be back in about 12 minutes.